Okay, welcome everyone. Today we are going to be covering connective tissue. So let's take a look at connective tissue and we'll look at the four different categories of, sorry, the four different types of tissues, that being epithelial tissue, connective tissue, neural tissue, and muscle tissue. And of these four tissues, I think the easiest ones to do are nervous and muscle tissue. And the two that are a little bit more challenging are going to be the epithelial and connective tissue. And the reason why they're more challenging is only because there are more categories that fall under it. Okay. And let's remember, how do we get to tissues? What is it that makes up tissues? Well, cells, right? Cells come together, a group of cells that have similar structure and function. When they come together, they're going to create tissues. What was it that came together to make cells? Well, that's going to be chemistry. They're chemicals: carbons, oxygen, hydrogen, nitrogen, carbohydrates, proteins, fats, etc. So tissues do have very distinct structure. They're going to have distinct patterns. Now, both of these, whether they're structure or patterns, that's form, right? And form determines function, or structure determines function. And when tissues come together in an organized manner that have similar patterns and similar structures and function, they're going to make organs like the heart and the kidneys and the lungs. Okay. When we study these tissues, we call this histology. Years ago, this probably 30 years ago, I remember taking a course called histology. And basically, it's like the anatomy and physiology of the cell. That's how amazing it is. It's just looking at cells, looking at their structure, how they come together, make tissues, and looking at all the different tissues of the body. Um, what an amazing subject. And I had a professor that was teaching it, and he just, probably as impressed as my students are hearing me with anatomy and physiology, that's how I was impressed with my instructor. How does he know all this information? Where does he keep it all? He had the whole textbook in his head. That's what we thought. And it was true. So when we look at epithelium, one of the things that I want you to understand about the contrast between these two, between epithelium and connective, is that when we look at epithelial tissue under a microscope, it's when we have a whole bunch of cells that can be closely packed together. And each cell, let's say, has a nucleus, right? So all these cells are closely packed together. Okay, whether it's square shape like I have here, or whether I make them rectangular shape like this, like tall columns, how these cells are closely packed together, or if I make them round and irregular shaped cells, how these could be closely packed together. These three different drawings that I made for you would be epithelium, when cells are packed closely together with very, very little extracellular matrix, which is the stuff that we find between the cells. Now, connective tissue, on the other hand, is when you have a cell here and maybe a fiber here, and then another cell here, and another cell here with a different shaped nucleus. So when you have cells that are far apart, with lots of extracellular substance or extracellular matrix, that's connective tissue. Okay? So we're going to look at epithelium first, and then after epithelium, we'll pause, and then we'll look at connective tissue. And then we'll do connective tissue, and then we'll pause, and then we'll get into muscle, then we'll pause and get into nerve. Okay? And then we'll talk about skin that has all four types of tissues within it. So epithelium is considered to be a lining type of tissue or a covering type of tissue. So when I think of a lining and a covering, what comes to mind is the skin. And the outer layer of the skin is called dermis. The upper layer is the epi. Epi means above. So epidermis is the covering of the body, but the external surface of the body. Now, epithelium doesn't only line and cover the outside of the body, but it can line internal surfaces as well. And in an earlier lecture, we talked about body cavities, whether it was the pericardial cavity, the peritoneum, or the pleura. We're going to have different types of epithelium that line those body cavities, and they also line tubules. What are tubules? Well, in the word tubule, we see tube. So anywhere in the body that we have something like this, where we have what looks like a tube, whether it's a blood vessel, whether it's another tube in the throat going up and down, maybe we'll call it the trachea or the esophagus, right? Anywhere we see tubes is where we're going to have epithelium. Now, if I were to put a slice through this and cut it and then rotate it, we'd be looking inside of that, right? We'd probably be looking inside and we'd see a lumen. I'll put an L here for lumen, where something's going to be flowing through that lumen. In this case, if it was a blood vessel, we're going to have blood that's flowing through there, okay? If I cut the esophagus and then we look at the esophagus through this way, we may have different types of cells that we find lining the lumen or lining the cavity or the space of the esophagus, right? Different types of cells that may make this up, okay? Okay, epithelium, also glands. Glands are classified as epithelium because they develop from epithelial membranes. Now, how do we name and identify epithelium? When we look at it under a microscope, we have to ask ourselves three questions, right? Especially if we want to differentiate epithelium from connective tissue. We have to ask, the first question is, okay, do the cells look like they're closely packed together or are they scattered far apart? If they're packed close together, like in that earlier drawing, well, that's going to classify it as epithelium. Then we can go to the second question that we have to ask ourselves. The second question we'll ask ourselves is, okay, when I look at this under a microscope, how many layers do I see? Do I see one layer or do I see more than one layer? If I see one layer, it earns the term simple. If it's more than one, we use the word stratified. So it could be simple epithelium or stratified epithelium. And we're looking at the apical layer, meaning if this is what we're looking at under a microscope, okay, and this is the superficial layer and this is the deep layer, and we have some cells here. This top layer is what we call the apical layer because the apex is the top. Okay, and it's also, this may be one section of this lumen here, right? So maybe we're taking this section out, we're blowing it up, and we're looking at it down here. So I'll put an L here and an L here, right? So here's the lumen, but once you cut it, maybe the lumen's up here. So we're looking at the apical, the cells that are at the apical layer or to the surface or closest to this lumen. So in this particular case right here, we saw that these cells are multiple layers. I'll put a nucleus, and all of these are okay just for funzy. So these are multiple layers. So it's going to be a stratified epithelium. But if we were to look, and let's say in this slice, and let's say here's the lumen, and let's say right here, this is what we saw. This is only one cell layer thick. So this would be a simple layer. So first question we ask, hey, are these cells grouped closely together or are they far apart? If it's far apart, it's connective tissue. If they're closely packed together like they are here, it's epithelium. Then we go, all right, is it one cell layer thick or is it more than one? Simple versus stratified. The third question is, what's the shape of the cell? If they are like flat and irregular shape, then we call them squamous. And if they look like a square with a nucleus and a cube, then it's cuboidal. And if they look like a building or a tall column, then they are columnar. So if we look here, right here in box number 1, these are flat, irregular cells. This is squamous. And the fact that it's one cell layer, we'll call it simple squamous epithelium. To the right in box number 2, here they are flat. Well, we asked ourselves a question. Are these cells closely packed together or far apart? They're closely packed together. Second question, is it multiple layers or one layer? This is multiple. If it's multiple, it's stratified. And then are these columnar shaped, cube shaped, or flat, compressed, and irregular? They're flat and irregular, so these are squamous. So this is stratified squamous epithelium. In box number 3, these are cubes, aren't they? They're squares, they're boxes. So these are cuboidal. And because it's one cell layer thick, it is simple cuboidal epithelium. Whereas box number 4 over here, these are cube shaped, but they're more than one cell layer, right? Here's one, here's 2 cell layers. So we call it stratified cuboidal epithelium. Now down below, in column number 5, we see that these are one cell layer thick, but these look like tall buildings with a nucleus. So these are simple, one cell layer, columnar epithelium. And what you're seeing up here that we did not see in box 1, 2, 3, 4, these are cilia. And these cilia can beat and move rhythmically. So if I really wanted to be specific, I would call this simple ciliated columnar epithelium. In box number 6, these are column shaped, but they're two or more layers, so it's stratified columnar epithelium. Now, off to the side here, we see something that is unique, right? It says it's stratified. Now, stratified means it's more than one cell layer. But we have the word pseudo there. Pseudo means false. False. So it's a false stratified layer. What gives it a stratified appearance, if we look at the nucleus, these nuclei look like they're at one layer. These nuclei look like they're at a layer, and then we have another one. So it looks like it's multiple, but in reality, it's only one cell layer thick. So we call this pseudostratified. And again, right here on the top, there are cilia. So I would call it pseudostratified ciliated columnar epithelium. Typically, we're going to have these cilia in places like the airway, in the trachea, in the windpipe, and we're going to see in the ovaries, in the fallopian tubes, and these cilia beat rhythmically. So let's say here is the trachea. I'm going to draw a trachea here. And right here's a tubule, if you would. It's a crude drawing. But what we're going to have here are these columnar cells. And these columnar cells are going to be on both sides, okay, lining it. And we'll put a nucleus next to each of these, and it's going to line the entire trachea. And then we have these cilia, these little hairs on both sides that come off like this. And we will also have, I'm going to change the pen color here. Let's change it to blue. Somewhere around here, there's going to be a cell and another type of cell here called goblet cells, and maybe another one here, a goblet cell. And what these goblet cells do is they're going to produce mucus. So it's going to produce mucus, it's going to secrete out, and it's going to stick to the cilia. It's going to leak out of the cell and then stick to the cilia, create this mucus lining. And what's happening is that air that you breathe in is coming down, right? It's coming down and in to the airway, and any dust or debris is going to stick to the mucus. And now the cilia are going to be beating upward. The cilia are beating upward to push the debris and dust particles up and out so that it doesn't make it into the lungs. Well, what happens in smokers, if you're a smoker and that smoke is coming in to the lungs, you paralyze and burn the cilia, and you damage these columnar cells. And when you damage these columnar cells, the body goes through a maladaptive process called metaplasia. Metaplasia. It's going to change from those columnar cells, and then they will be replaced by squamous cells. And if you've ever heard of squamous cell carcinoma, that is cancer. We have to be very, very careful. And typically, this metaplasia is when someone wakes up in the morning with that smoker's cough, where you hear them going, right? They have to force, because those cilia are not there. They're trying to get all the tar and all the debris and all the carcinogens up and out of the lungs. So again, to review, we have simple squamous, and then down below we have stratified squamous. This is the deep layer, and up here is superficial. So what we have are several layers of those flat, irregular cells, stratified squamous epithelium. Here we have simple cuboidal, and here is stratified cuboidal. Here is simple columnar, and here it looks like it's stratified, but it's really not. It's pseudostratified columnar. And one last that I'll mention is called transitional. Transitional epithelium is when cells alter and change their shape based on whether the organ that they're surrounding is distended or not. So think of a balloon. If a balloon is deflated and you put a square shape with a permanent marker on the outside of the balloon, you could see the shape that you drew. But once you blow it up, it alters and changes its shape. It's transitioning. So we have transitional cells. Okay, typically you will see them around the urinary bladder because the urinary bladder is like a balloon. It can be inflated or deflated. So the purpose of the epithelium is primarily for protection, absorption, filtration, excretion, and secretion. And then they act as a sensory receptor, especially on the skin. And that's what allows you to tell whether like an insect is crawling on you or a mosquito is on you. But if we think about the skin, the lining of the skin of epithelium, its many layers of the stratified squamous epithelium, it protects us against excessive friction or a bacterial invasion or from spilling chemicals that could be very harmful to the body. We need multiple layers of protection there. We also saw in the previous picture that we have ciliated epithelium. And I drew for you the trachea or the windpipe, and we'll see cilia in the respiratory tract that can sweep dust or other foreign particles away from the lungs. But you'll also see it in the ovarian tubes or the fallopian tubes. And that's what's going to try and sweep the sperm to the egg for fertilization. Okay, let's see. Absorption. Well, throughout the GI tract in the stomach and the small intestine, we're going to have epithelium. And think of absorption, think like a sponge. A sponge can absorb water. So epithelium within the stomach and the small intestine is absorbing nutrients to bring all of those vital nutrients into your blood vessel to drive it into the cells and drive it in for energy. Filtration. When I think of filtration and excretion and secretion, all three, I actually think of the kidneys. But the kidney tubules, you see, absorb, secrete, and filter. And when the body is filtering blood, it can pull out debris. Everything that it wants to get rid of, it will excrete from the body. Okay? The kidneys can also secrete things. The kidneys can actually secrete certain hormones that can have impact on the body. It can have impact on the bone marrow, especially erythropoietin. It produces more red blood cells. Absorption, well, maybe your kidneys want to get rid of calcium, or maybe it says, hey, this person's in a fasting state. We don't want to get rid of calcium, or we don't want to get rid of all this glucose. Let's hold on to it. Okay. So epithelium has quite a few functions. We talked about cilia. Those are those long, slender extensions that come off the cell membrane. They help move secretions. We spoke about that one cell, and we called it a goblet cell. A goblet cell produces mucus. Okay? Microvilli are very similar to cilia. They're just a whole lot smaller. They're much smaller projections. And again, these help to increase the absorption. So we're going to find microvilli throughout the GI tract, the gastrointestinal tract, to absorb nutrients. So I've seen this show up on some quizzes before and some old exams before, and it's kind of like a drag and drop or a matching section, if you would. And we're trying to figure out, well, where does simple squamous go with, and where does simple cuboidal go with, and how about stratified squamous, and then the pseudo stratified, and then transitional. Well, remember transitional, I mentioned like the balloon that alters in shape, or the urinary bladder, depending on whether the organ is fully distended or whether it's relaxed. It can change in organ volume, so that's transitional. And pseudo stratified columnar, this is where you'll find these throughout the respiratory tract. And that's where they have those goblet cells that produce mucus. This is the type of epithelium that functions in movement of secretions like mucus, secreted by those goblet cells, and they move them across the apical surface. So that picture that I drew before that had all the columnar cells and had the goblet cells, typically you'll see them as pseudo stratified columnar, false stratified. Simple squamous, we'll find these in the lungs, right? So if here's the windpipe, and then here's the primary bronchi, and then you have your secondary bronchi. Eventually, it's going to go into the two lungs, crude drawing, and it's going to bring in oxygen, again, crude drawing. So deep, deep within, we're going to have something called the alveoli, where we have one cell layer thick, that's it, one cell layer, so that oxygen can easily pass through. And that CO2 can go the other way, right? So oxygen comes in, and then CO2 can come out. And we want simple squamous, just one cell layer thick of those squamous cells to allow things to diffuse through. Simple cuboidal, we're going to find these in the kidney tubules, especially the proximal convoluted tubule and the distal convoluted tubule. When you look up the proximal and the distal convoluted tubule, these are part of the kidney called the nephron. The nephron, which is the functional unit of the kidney. That's where we're going to have simple cuboidal epithelium for absorption, secretion, and protection. Stratified squamous, I think of the skin. That upper layer has stratified many layers of flat, irregular shape for protection of the underlying layers. Okay? So again, we name epithelium. First question, we have to ask ourselves, there's three. Are these cells closely packed together or are they far apart? If they're closely packed together, it's epithelium. Is it one cell layer thick or is it multiple? If it's multiple, it's stratified. If it's one, it's simple. Is it flat, irregular shaped? Then it's squamous. Is it box-like or cube-like? Then it's cuboidal. Does it look like a tall building? Then it is columnar. Again, the cuboidals we said we would find in the kidney tubules, where we have the proximal convoluted tubule and the distal convoluted tubule. All right. Squamous cells for protection, secretion, and diffusion. Protection will probably see more than one cell layer. We'll see stratified, or diffusion will be simple. We'll see one cell layer thick. Pseudostratified, we said the cells appear, especially those nuclei, are going to appear at different levels, so it's really not a true stratified. It is a false appearance of being stratified, and oftentimes where we see pseudo stratified, we're also going to see cilia. So it'll be pseudostratified ciliated columnar epithelium in the trachea. Transitional, this is the one that, depending on the organ size, like the urinary bladder, is it distended or not? The ureters, the urethra, these parts can stretch. They can alter in shape, so they could be flattened cells. They could be more cuboidal and round. They change their shape depending. So when we look at the body and we look at the different parts, we can see, well, in some areas we're going to have pseudostratified ciliated. There we go, pseudostratified ciliated columnar. We said we're going to find them in the respiratory tract, and that's what this is. See, the nuclei are at multiple levels, but yet it's only one cell layer thick. So it's really simple. but it has this appearance of being stratified. How about here? Look at this. Here's the apical surface. So this top portion right here, this is stratified. So it's stratified squamous, flat, irregular. We could see this in the skin. We could see it in the esophagus. Any place that's coming into contact with something from the outside world that could potentially irritate those cells. Simple squamous epithelium. Well, remember I said in the lungs, deep in the lungs, we have something called the alveoli. Well, those are the air sacs of the lung. And we only have one cell layer thick here. Okay. So simple squamous epithelium, alveoli of the lungs. In the kidneys, simple cuboidal epithelium, especially at the proximal convoluted tubules and the distal convoluted tubules. Make sure you look that up. You know, it's good to kind of get a visual of the nephron. You're going to hear words like glomerulus and loop of Henle and collecting ducts. Really quite a fascinating part, the kidneys and that nephron. Kidneys have millions of nephrons, by the way. And then in the intestines, we're going to have simple columnar epithelium. So to summarize, when we look at epithelium, again, three types. It's either simple squamous, simple cuboidal, or simple columnar. On the multiple layers, on the stratified, we have stratified squamous, stratified cuboidal, and stratified columnar. And then the two outliers, which would be pseudostratified columnar and transitional epithelium. Okay, good place to take a break here.